All right. Welcome back, everyone. I can see the numbers of people calling in are slowly ticking up, but given that we are together for only an hour, I think I'll go ahead and get started, especially since the opening part is just review, and people calling in can hopefully follow along where they may before we sort of move on to the new materials that we'll be talking about. Just to reintroduce myself in case there's anybody new this week. My name is Fred LaPolla. I'm a librarian here at NYU Langone, and we'll be continuing Introduction to R, picking up where we left off today, talking about indexing. If you're not sure what that is, don't worry about it just yet. But right now I'm gonna go ahead and just do some quick review, sort of hopefully to iron out or remind you of things we talked about last week. And so this will be sort of a quiz. So if you can go to menti.com and use this code 487717, we will do some review. So you may remember from last week, we talked about assigning variables, and who remembers what symbol we used? I'm seeing people calling in, or dialing in rather. This is excellent. Yes, so the 100% of people have it. It is this arrow symbol. You can also hold option minus or option hyphen, and you get the same symbol. The other symbols we see on the screen, 2 equals will mean that we're saying in a sort of a logical way, if we're trying to test a condition or trying to filter a set of data, if we're saying, is some value equal to, is some column equal to, for example, 40 years of age, we would use the double equals. This percent symbol with the percent arrow percent, that can be used in certain packages, particularly in the TidyVerse family of packages for sort of carrying things along or piping things. Haven't really talked about that yet, but you may see that going forward. An exclamation equals means not equal. Some people will just use a single equal sign for assigning variables. The convention in R is not really to do this, and instead to use the arrow. The equal sign can have different meanings and different functions, so that tends to be a good reason to sort of use the arrow rather than the equal sign. Okay, so feel free to type in a command you would use to load a CSV file, which again is that very low tech Excel file or spreadsheet file, into R. Great, first to have it, read.csv. And that's correct, so I'm not going to wait for everybody to enter that, but I'm glad to see people have that down. And what symbol would be used for writing a comment into your code? So if you're in the scripting pane, you can add this to avoid having, what's the word I'm looking for? The line be read by your code, or by your code. So yeah, so it'll just be sort of human readable. Do want to really encourage you to do this. Even if you think or feel at the time that what you're doing is very clear and later you'll understand exactly what you're doing, typically this is not what happens. Those of us who've worked with R or other languages may recognize the frustration of having not commented our code, come back, a month later and said, what am I even looking at? And you can sometimes start at the very beginning and read through and figure it out, but it's not a very efficient or fun process. So I strongly recommend using this hashtag when you want to comment your code. So every few lines, just sort of say, this is what I'm doing. It does not have to be fantastic prose. It just has to be something to let you know for later what you're doing. And so we talked about some ways to get a general summary of what we're doing. So what were some of those commands when we pull in a data set? So you might remember from kind of the end. Great, so head, so that gives you the first 10 or so lines, but you can actually set how many lines. Summary, head, great. View, if you want to see the whole thing. Describe, which I believe is in the psych package. Let's see, what are some others? Head, tail, STR for getting the structure. Great. So it looks like the lessons of last week have, oh, this is actually sort of skipping ahead. So I'm going to jump back to my materials from last week, where this is sort of where we left off, and just talking about some of those summary options. So here we had the pastex or package with their stat desk, and you can get nice things from that as well. I have this around because it was giving in scientific notation. One other thing which I neglected to mention is for today's purposes, if you have questions, definitely feel free to put them in the chat as we're going through. So if anything I'm saying is unclear or you're not sure about something that's happening on the screen, definitely the best way to do this is to go to the chat option. There's also a Slack for this class that should have gone around for the link to join. So hopefully you'll be able to join that, as well as the class Stack Overflow for posting and asking questions. So NYUMC coding courses is a place to ask those questions. Excellent. So yeah, I'm just seeing in the chat, there is this link to access the materials from SharePoint. Something I just learned about Zoom is that people who join after this point will not be able to see previous chats, unfortunately. So it may require, we may have to actually post this a few times, which is something I didn't know at all before yesterday. Okay. So now to really jump into today's topic, or the first of several topics actually that we'll be talking about, is a concept called indexing. So you may recall last week while I was working in R, I would frequently refer to columns of our NYC Haynes data frame using a $sign. And that is one way that we can index or reference a specific part of our data within R. And indexing is just the general term for this idea of accessing specific parts. So one option is this $sign, and that can call up a whole column of material. So if we said mean NYC $sign cadmium, I guess I'll actually just go ahead and do it. Pulling up R, I'll do it in a the same blank, or perhaps, let's see, if this untitled R document. So if we said, let's say mean NYC cadmium, not sure if there's any NAs in there, but I'll say NA.rm equals true. It will tell us down here at the bottom, I need to move this up slightly, 0.421. So that is one way we can reference a specific area of our data frame. Another option is to write the name of the data object, and then if it's multidimensional, and so by multidimensional, I mean it has things like columns and rows, we can give it first the row and then the column separated by a comma. So we have the option of doing more than just one number. We don't have to only reference individual cells. So this here would be accessing the 4th row, 5th column. So if I pull up, for example, this is just a spreadsheet. We haven't pulled this in yet, though we will be working with this later. That would be going to the 4th row and 1, 2, 3, 4, 5 over. So that would be a way of communicating this cell, for example, if this were the NYC data frame. We can also use, you may recall from last week, we talked a little bit about using colons to mean every number between a set. So if I pull R back up just to review, if I write 1 to 10, I get that written out here. So if I wanted to reference, for example, the first 10 rows, or I can even just say NYC, let's say I want to do the first 10 by 10. I can do this in here and hit enter. And now when I go down, it's telling me that I have this is, we can see 10 rows by 10 across. Let's say, so I see there's a comment, which maybe there may be a need to pull in the data frame if you want to follow along. So I'm just going to copy and paste this into the chat in case this is the issue. So try running that if it is the case that you're not seeing NYC up here. Because again, I apologize, we are sort of jumping in midway from last week. So we also have the option, we could pull an entire set. So if we left either the row side or the column side blank, what that would be doing is saying every row or column, depending on which side we left blank. So if we said NYC 1 through 10, comma, and hit enter, If I go down, it's a little bit hard to view here, but we can see we have every column for the first 10. Oops, apologize for difficulty navigating on this menu. So we have 1 through 10, first couple of rows, going further down, 1 through 10 for the next ones. And similarly, if I did comma 1 through 10, I'd be invoking every row, but just the first 10 columns. So that is the general idea of how we can use this indexing to reference specific things. They also do not have to be sequential. Whoops. So if I said instead, for example, C 4, 5, 6, comma, C, and C, if you recall, stands for concatenate. I'm not sure if we talked about this last week. C is a way of grouping things that are perhaps non-sequential. So it's just saying, take the 4th, the 5th, the 6th. Those are actually sequential. So let's do it. Something not sequential. 4, 5, 9. And let's say we did, for the sake of argument, 2, 5, 14. And I run this. It will tell me, so this is column 2, column 5, and column 14. And why is it giving me, oh, and then, sorry, for rows 4, 5, and 9. So it's just this small section here. Let's see. So yeah, so C is a command within R. It's almost like an annotation. It's very seldom used as a standalone. It basically means group the things in here if they're not sequential numbers. So we could do 1 through 10, and it just reads that out. But let's say we wanted to assign some numbers to a variable. So let's say we wanted to do 4, 9, 33. If I just hit enter, it says there's some error. It doesn't understand because it's just a list of things being given it. Whereas the C, if I do that, now if I run X, it gives me those same numbers. So it's basically letting R know, expect a group of things, more than one thing, to be passed along that are not necessarily sequential. So if you see that as you're sort of going along in R, just be aware that that's what it means here. So we're saying, because here it could accept the colon, but here it cannot. It really needs some way of being told to understand this. There's also the option, we can actually delete a column. I'm not gonna, well, actually, yeah, why not? I can go ahead and do it. So let's say I realized I do not want the first column. Let's imagine, so the first column right now is just sort of a general subject ID type number, but let's say I just wanted to get rid of that. I wanted to give somebody a totally de-identified set. I could do a negative sign, so I could do NYC -1. And what that's going to do is that's going to delete the first column. So in this case, the symbol is, yeah, -1. So we can see over here in our global environment, which is this area of storing all of our objects that we're working with in R. There's currently 23. If I do this, it actually printed out the whole thing in here, which is, not terribly useful to me. What happened? Why did it not? Now it still says 23. Oh, sorry. I need to, so it's just printing out without the first one. I would need to read it back into itself. So now it's down to 22. If you are me working in R, you will often make those sort of simple mistakes and realize, oh, I need to go back and do things. While it may seem like I might be just covering for having forgotten to type something, that for me at least, and I suspect for many others, is a very common step, is some typo, some small error. Oh, I have to do this again. So I just want to sort of underscore that that really is a major part of working with R. So yes, this is just illustrating the similar point. You could also, if you wanted, use this to reassign or write over a cell. So you may or may not have reasons where you might want to do this. One typical reason that this might be an issue is, especially for those of you who will be doing work with matrices, so gene expression work, RNA-seq analysis type work. Let's say a cell has a typo, a stray letter got typed in while you were working with the data. Maybe you got the data back, you submitted your samples to be processed, you got it back, and when you looked at it, you accidentally typed something into it. You could, of course, and you didn't realize until you read it in R, you could technically go back, correct it in the spreadsheet, and then reload it in. But if you didn't want to go through all that, maybe you're on a different computer, doesn't really matter. You can assign a new value to any given cell. So we could imagine if, let's say, NYC, 4th row, 5th column, had, instead of just the number 5, it had 5 commas or 5L or something, like some small error had gotten into it. We could assign it over with just the number 5 using that assignment, and now it will read that over it. Just glancing over at the chat. And yeah, lots going on in the chat, so apologies to those encountering errors as they go through. So yeah, so you can also create a new column if you wanted using indexing. So you could, for example, in this case on the screen, what I was doing is, let's say we wanted to get a cholesterol ratio, so something that's done sort of for clinical purposes, perhaps on your own. If you get blood work done, it will tell you the ratio typically. And my understanding is that it's done by dividing the total cholesterol by the HDL to get that ratio. So what we could do is we could go over, and let's go ahead, why not? And I could say NYC, so currently there's actually only 22, so we could say, for the sake of argument, 23. I can assign into that currently not existing column, we could say NYC dollar, if I start typing cholesterol total, divided by NYC dollar, what was it, HDL. And when I hit enter, it doesn't look like much has happened, but if we look again up here in the upper left, now we have 23 variables. And if I wanted to do summary NYC 23, it's telling us some basic information. So I do not recall what is considered a high cholesterol ratio. And again, for today, it doesn't actually matter. So it's really just to illustrate that we could do that. We could create a new variable by using indexing, just sort of plugging it onto the end at the 23rd position, as it were. If I wanted to get rid of that, I can just do comma, -23. And, oh, whoops, same problem as before. Just gonna assign it back. NYC, we're back to our 22 because we don't have our first column anymore, though we will be pulling that back in a little bit. Let's see. Oh yeah, so let's try out another Mentimeter question just to sort of quiz you all. Oh, well, so some people have already answered, but those jumping ahead. So if we want to look for the 3rd row, so horizontally down, 4th column, 4 over. So if it was that Excel document example, so here by here, so 3D as it were in Excel speak. Excellent, and it seems like most are getting this. So yeah, just to sort of drive this home, this is going to be this 3,4 to access that. If we wanted more rows, we could do 3 colon or C, and then in a set of parentheses, 3, 7, whatever, and so on. If anybody is not seeing the questions, it may be that I would say look at what's on my shared screen. I'm not entirely sure, but it may also just be slow because I'm not sure it was really designed to have, looks like we have 158 on the call. So it may be struggling with a large number of people. Oh yeah, so I'm actually gonna move on to back to the slides over here. So something which we didn't talk about, but will be very important going forward. Actually, before I sort of move on from indexing, I should open it up just for a second on the chat. Are there any questions about indexing that I did not raise? So not so much if you're having technical trouble at the moment, but more conceptual questions, or if you saw something and you're not sure why I did it, if you could tell me, let's see, or ask a question. If you delete a column, there's not really an undo, to be honest. So One of the nice things in, let's see. So one of the nice things in R is we're not actually interacting with the raw data, which I have been pulling directly from GitHub using a command that began R curl and then pulling the URL. But something that actually you do kind of want to be aware of, which can be a little bit frustrating at times, is that when you make changes like that, there's really no undo button. There is actually an undo in our studio, but that refers to typing issues. So if I wrote right here, NCY, and I wanted to undo, I could do edit, undo, but that just undoes the typing. It doesn't undo commands to my actual data. Fortunately, when we have a lot of code, especially if we have code that's sort of reading the data in, we could rerun to get up to that point pretty quickly, like rerun all the commands up until then to get there. Let's see, just reading back through, when you did NYC 4,5, the output was 1, 5. The 1 is basically telling you which sort of object in a vector. We'll be kind of talking about that going forward. So let's say there was more than one thing. It would say position 1, there's a 5, position 2, there's something else, and so on. But here, because we're just working with a very simple cell, it's saying that the only one is the 5. There's a question on na.rm. So na.rm stands for na.remove, or remove NAs. R is not very savvy about knowing what to do with NAs in numerical fields. So if we try to take the mean of a column or anything that involves an NA, it will say error because it ran through doing the the summing and the dividing that you do with an average, but then it would sort of break and turn off and say, we can't finish this. We don't know what to do about NAs here. So we're not going to run the mean. So na.rm with this numerical is just remove the NAs, ignore them when you take that mean. That way it will just give you the mean, not including those NAs. Let's see, I'm just sort of looking through, seeing TAs answering things. How would you compare one column to the other? Okay, we'll be talking more about that. That's sort of, not to gloss over that, that will be definitely, I think, something that will come up. There's a few different sort of ways that one might do what might be more of the analysis of our data. And so depending on the nature of it, some of the things I'll talk about, but probably even more the things the experts will be discussing, gene analysis will be talking about, but we will be talking about some ways you can get kind of basic information, such as comparing if getting a quick sense in an exploratory way, as well as a more numeric way. Does that mean you have to code non-numbers in your spreadsheet as NA? That is something which I would have to think about if you have to code them going in, i.e. not a number. That, to be honest, I'd have to double check. You could also, my understanding is, bulk sort of recode certain values that you've pulled in, so you don't have to type them all. But you could say if there's a value that say it was 999, you'd set for missing or for NA, you could then pull that in. Let's see. And I think that probably a good option for knowing how many items, probably something like length. But again, I would have to sort of play around to double check what works best. So off the top of my head, something like length would probably be a good option just for seeing how many values. Also, some of the summary ones will tell you, depending on the type of data. So hopefully that's somewhat useful in terms of my answers. I feel like I've been rambling just a little bit. But if anything I've said isn't clear, we can also discuss this in Slack later or on Stack Overflow. So sort of jumping back, so sort of, actually that's a nice segue. So there's a lot of commands that we'll talk about, and there's way too many to memorize. A colleague of mine has who teaches a workshop on Python, and who's actually a web developer in the library. He has a nice slide that he found, or a nice screen grab of someone on Twitter who's actually one of like a higher up at Google, saying that every time in Python they need to look up the length command, they have to Google it again, because it's just, these things can be often very small and hard to remember the exact phrasing, and often for things like length, which happens, I think, to be length. Let's actually check. Is it length? Let's do length, NYC dollar, sure. So it is length, but perhaps in a different system, it's LEN. So it can be easy to forget. So there are good, it's always good to just remember that you're not doing this in a vacuum where you're expected to have it memorized. First off, one thing to really note, R is case sensitive. So perhaps I should have led with that. So people have been encountering problems around typing. Case really matters. Spacing, not so much. So you could have, in the case of, for example, if we did the mean, I could add a bunch of spaces and it shouldn't really matter. Yeah, we still get it. But if I had written NYC with a capital N, it's going to give me an error. So it's really important, especially if you're seeing errors, just to double check. Maybe there's a typo with capitalization. It's not really good at intuiting. So if there's any sort of typo, any spare commas, any spare symbols like a bracket, or maybe you had shift held, so you used a curly brace when you wanted a square brace or vice versa, it's not going to be able to figure that out. So really, I mean, when we've done workshops in the library, the #1 issue by far that people come along into is hitting typos as you're going through. Also, sometimes when we're coding, we may be left, especially when we've done a more complex command. So far, we've done very basic sort of functions, like just where there's one set of parentheses and it's the mean of whatever. But sometimes when we have a lot of nested arguments, we may be left with just a blinking plus sign, just to illustrate exactly what I'm talking about. Let's say I do, for the sake of argument, I'll just again do a very basic one, but I'll just do the one above. So I'll do this. But let's say I missed closing a parentheses. We'll be left with this plus sign. And I realize it's a little bit small, although I guess it may look different. But down here, we have this plus sign. And in this case, we could just close the parenthesis, easy enough. Oh, and it has the error because the NYC was capitalized. But in my experience, generally when this happens, it's not that we've forgotten the last parenthesis, it's some complex issue along the line. And the easiest thing to do is to hit the escape key. So if you're left with an error where you just have a plus sign, it's not sort of ending and bringing you back to this, this arrow symbol, which lets us know we can do new coding. Try hitting escape. It'll sort of cancel out what we're doing and bring us back to the start. It won't delete the existing things, so we could copy and paste them and add a closed parenthesis or double check that all of our parentheses and quotation marks all match with one another. So if they open, they close. But if you see the plus sign, best bet, hit escape, and it'll just sort of bring you back to this blank line. When in doubt, Google it. So I saw in the chat there was some discussion of issues on how to find certain things. Hopefully through the course of taking this class, you'll learn some of the sort of the jargon, so it's a little bit more effective in doing searches. But in general, you'll need to look things up. So one very commonly used tool is definitely Stack Overflow. So Stack Overflow, I have looked up, there's also a related phrasing. I think it's the same organization called Stack Exchange. I once knew what the difference was, but from a, from a practical standpoint, where we're not too concerned with their overarching, corporate mission or whatever, if somebody says Stack Overflow or Stack Exchange, we're basically talking about the same thing. So for the class, of course, we have a class instance, but if we wanted to just sort of search everything, we could say, mean in R, probably not actually that many interesting things, but here we can actually see, I'm not sure which of these are from NYU and which are not, but we can see, well, this is from 2014, so this is pretty old. A lot of different questions. Normally, we're going to want to be a lot more specific than something vague like mean in R, so we're going to look for exactly what we're doing. We could also use Stack Overflow if we're running into an error. Then we could go here and sort of look to see, what does this error when I'm, plotting a histogram, I get this weird error and I don't like it. What does that mean? And often, I would say 99 times out of 100 or 999 times out of 1,000, somebody out there has encountered a similar problem to you before, and you can kind of figure out an analogous solution for your data. So I really like Stack Overflow. It's basically a giant message board. It's not just for R. So if you're also in the Python class or you're interested in other tech topics, it's a great resource to be aware of. Another is called Quick R, although the URL doesn't quite match. Oh, I have to, that's fine. I should have done that in a new tab. This I really like as a tool. I guess I've happened to default to the frequencies. I like this for kind of what I would think of as bread and butter type issues. So let's say I wanted to know in R, how do I run the analysis of variance? I can go in and it will give me the information in a pretty straightforward way. In contrast, the information in Stack Overflow is users responding to questions others have posted and sort of upvoting and downvoting based on how useful they are. So it can be a little bit of a fishing expedition that you're just kind of reading through and trying to figure things out. Whereas this statmethods.net can be really nice in terms of finding kind of bread and butter walkthroughs on normal, frequently done sort of statistical forms of statistical analysis. So if you just Google quick R, that should come up and it can be nice. So also I'll note that sometimes the things we want to do may not come with R right out of the box, or you could do them, but they'd be more difficult. A nice feature are these things called packages. They're basically sets of arguments. So often when we're looking either in Stack Overflow or perhaps on our bloggers, we'll see commands that are using different packages. So previously we used the psych package and the paste ECS package, as well as rcurl for reading in the URL from GitHub. So these will be just sort of sets of commands somebody has written that will hopefully make life easier. Last week near the end on the chat, I saw there was a question about sort of something comparable to MATLAB. My understanding is kind of a centralized, controlled place for these packages. Whereas R, because it's open source, is a little bit like the Wild West. So it's good to sort of see, is the package I'm going to be using, is this widely used? Is it kind of rarely used? Has it been maintained? And these are things you can kind of figure out based on how much people online tend to be talking about them. Some of the packages out there will be very frequently used. There's a set called the TidyVerse by Hadley Wickham. It's actually a set of packages, so it contains all of these packages within it. And to be honest, several of these I haven't really worked with too much myself, but ggplot, dplyr, very commonly used ones. So let's go ahead and on our own. I have the repo typed in. You might not even need that, but let's try installing a package and then loading it. So I'll do that on my computer. It might actually request me to restart things since it's already there, but we can see if it works. So a package is a file that consists of different functions. I'm not really entirely sure if that answers the question in the chat. And to be honest, from sort of the practical standpoint, the basic thing to understand about there are often data files associated with it. The practical thing to understand about the packages is they allow you to use commands that might not have been available otherwise. So if I want to try installing this one, I'll do install.packages and then in quotation marks, TidyVerse. And I'm going to hit run. Mine's saying it's already there. So sure, why not? Hopefully this doesn't make huge errors for me. Famous last words. Yeah, and then the note in the discussion, to actually get it to run, you're going to then need to do library, quote, or open close parentheses, TidyVerse, no quotes. So something important to note is once we get this going, hopefully I didn't just make a huge error, because there's no sort of central body within the R universe, because individuals are writing these and creating these, different packages may have the same name for different functions. So what I mean is an example here would be in this one that we're creating in something called dplyr, which is part of this TidyVerse set of packages. There's a function called filter. And what that can be used is we could filter down values in our project. So let's say we had we wanted to, for example, do, what's an example? So let's say we only wanted to look at the youngest age group. We could run filter of our data frame, and then only filter those out. There's another filter though that comes out of the box with base R, and so they mask that, or they basically turn that off. So you'll only be using the filter of the package we just turned on. So it's a lot of explanation just to say, when you run a package, so if I do library TidyVerse, let's see, I have the stop sign, which is letting me know something's happening. So yeah, warning, this was built on an old one. Often you'll get these red warning messages. They're typically nothing to really worry about. If you get an error message, it probably stopped things somewhere along the line. But yeah, actually, so in the chat, I see there are things where it's saying it's masking certain commands. So it's masking things from this package called psych and masking things from a package called stats. So filter, actually, we can even see the example. I don't really remember what stats filter even does because I'm more used to using dplyr filter. And it's just saying it's going to use sort of this top level one on the left of the chat, and not the one that's been masked. So it's using the one that you've sort of loaded most recently. So the take home, that's a lot of explanation just to say, when you want to use the commands of a package, you need to run library, and then the name of that package, no quotation marks. Okay, so yeah, so here's just an example. We're gonna be talking about filter and the other tools that come with the TidyVerse in a little bit, and a few probably in upcoming classes, probably won't get there today. But just to illustrate what that does, we can make subsets of our data much more quickly than would have been easily doable without having used the package. Often there is another way, but being able to use these packages can be really nice for quickly creating subsets and creating transformations to our data. One thing I will note, so I suspect this probably isn't a huge issue today. Sometimes things will, at least in my experience, I've had issues where downloading the packages can be a little bit slower when I'm on campus. So that can be a problem. And sometimes it does require playing around a little bit with it. One option is you could go to, let's say I wanted to get this, but it wasn't working directly. You can go to TidyVerse and just search the name of the package, CRAN. Let the wheel spin. You can see I've done this before for demo purposes. So here, CRAN project package is TidyVerse. And you can actually download, if you're on Windows, a zip drive, or on Mac, you can download this TGZ, which is sort of a Mac specific, very similar file. So you could download that to your say your downloads folder. And then in R, if you wanted to manually do this, you could go to packages, install. So the default saying go to that website, that CRAN repository. You can actually download it from your computer, from the downloads folder, and find that file and open it here. So that's another option, which sometimes when I'm having a lot of errors, that can be a convenient sort of workaround. So again, it's go to CRAN, search the name of the package, and CRAN, C-R-A-N, download that package there for your operating system, and then go to this packages, install, switch from CRAN to package archive file. So here, because I'm on a Mac laptop, it's .tgz or tar.gz, and then I would just find the name of it in my computer. Okay, it's even popping up. But I'm gonna say cancel because I don't actually want to do that right now. But just another option if kind of your default install.packages isn't working. Okay, I'm gonna skip that. So these are just some questions. We'll definitely make sure you have these, but if you want to sort of explore these questions on your home computer or on your time, that should be a good way to sort of cement some of the understanding of R. Okay, so we're going to sort of change gears. We're going to just basically dip our toe into data types and structures in the next 20 minutes and then resume this discussion on Thursday. But before I do so, aside again from sort of technical issues, and problems installing packages. Are there any questions about packages? The other thing, actually, I'll note, and we'll talk again more about this in coming weeks, but you, especially those of you who are going to be doing work with a set of packages from the group bioconductor, so it's very commonly used for bioinformatics work. It's actually easier to go to their site. So if I go to bioconductor.org, I'll just sort of go up a step. Whoops, what's it doing? If I go to bioconductor.org and I want the R packages, they have a whole bunch. They actually have code there that's better to copy and paste into your console and run their code rather than the install.packages command. Basically, some of their commands work only with specific versions of R. So in my experience, getting them to run properly, you're better off just going to bioconductor.org and looking for their packages. So if we go in, we could see a list of their various packages. Let's see. So if we just click, these are some of them. Let's actually get rid of all these things. But again, this will be less of an issue for this portion of the class, but something to be aware of going forward, and in particular for the more bioinformatics portions that you'll be moving into. But there's a lot of these options. Some may be familiar, some may be not familiar, but just be aware that bioconductor.org can be a great place to go to. Let's see. Is there a command to update packages that you've installed before? That's a good question, and I would have to look into that. I don't know off the top of my head. Yeah, so when you want to run a package, you can say library package, and it will stay on for that session. You may then have to make decisions if you load another one that masks that package. You may want to consider that. Okay, so I'm going to jump over to talking about data types and data structures that exist within R. And I'll say that this topic is sort of at the same time maybe a little bit abstract, but also kind of very fundamental. And in particular for doing bioinformatics work where you're going to be working with a lot of matrices. So for example, if we pull up our spreadsheet that we're not really working with just yet, but if we have these gene expression information for these different wild type and knockout mice, we've got what's called a matrix here, what will eventually be called a matrix. And we want to be able to do analyses that are going to be treating all of these as numerical data. So it will be important to know what types of data you're working with, and if, as is typically the case, what R pulls in your data is not the necessarily correct type of data for you to do your analysis for your work, it's important to be able to change the type of data and change the structure of the data so that you can run your analyses without any problems and with it doing what you expect it to. So yeah, that's just a long way of saying, while this may seem kind of abstract, I do really think it could be very important for bioinformatics work. So I'm going to skip the review because we just talked about that. But yeah, so hopefully by the end of this section, so it's not really going to be the end of today, but this session and next session, you'll be able to identify the different data types that R works with, name and explain the data structures that are available in R, and you'll be able to look those up as well as change them. Typically, when we pull in a spreadsheet, it'll be pulling in what's called a data frame, but often, especially for bioinformatics work, for example, creating a heat map of gene expressions, you're going to need to change that because it's not going to want to work with a data frame. So this is still the same set of data, so we don't need to pull anything new in. And I'm pulling this in, again, because it has different types of data, and I feel like it works nicely for seeing the different things that are available. So just to quickly go through the options that are here. So to list off the main types before diving into each of them and talking about what these mean, there'll be character data, so things like text that are not sort of groups, but just like, let's say you had a spreadsheet of members of your lab, The names of everyone in your lab would be characters. It wouldn't make sense to have them be groups, probably, but they would just be sort of written out in plain text. Numeric and integer are kind of what they sound like. So if it's a number value, the main difference is that integer will just sort of be the rounded off number, and it won't store that extra data. So in some cases, you may want to really think about the efficiency, but in general, probably my sense is that numeric data will be fine. What's called factor data in R, and this will mean either categorical or ordinal variables. We'll talk a little bit more about what that means in a minute. Logical variables, which just means if it's true or false. So if you were to write true, if we were even just do that right now, you'll note that it writes it differently than if we wrote, for example, well, actually I'll do it up here, because I think it should do it up here. So true, it turns blue. If I just wrote my name, it stays in the black text. It doesn't really recognize anything about my name, but true, it turns blue because R knows that true means something. And we'll talk a little bit more in a moment what that means and how it can be used. And it can also handle complex numbers. I personally have never worked with complex numbers in R, or actually at all since high school math, to be honest, maybe college math, but this is not part of my, my research life. So it may be relevant to you, but something that you would need to explore on your own later on. So I mentioned character. Perhaps very intuitive to think of it as things like a name, text written out. Something to note is often when we have a column of data, so I'll actually pull in my R data again, just so I have all of because I'm down to 22, I want all 23. Let the wheel spin. Well, that goes, oh, it's because I'm gonna hit stop. Well, okay, so it's there, and it's there. So a command that will be useful to us is the class command. So we can see that first column, NYC, I'll just do comma 1, and it's a factor. But if we go back, if I do view NYC, you may recall it's a key. I'm not really sure it makes sense to think of this as a factor. They should all be unique, otherwise it's not much of an effective key. So we don't really necessarily want that to be a factor. And we'll talk in a little bit about how we might change it. So it's really good to just note these things right off the bat. So numeric data, again, is going to be numbers. Something important to note, again, is just because it looks like a number doesn't mean R read it in as a number. And this I've seen come up specifically in the context of when working with matrices, which we'll discuss a little bit more what I mean by that in a moment. But let's say you're trying to make that heat map of gene expression data, but somewhere in your spreadsheet, somebody had a typo, so there was a letter. It would think that all of these, even though they're written as 7.25, 6.056, it would think that they were text. And a giveaway that it's reading them as characters rather than numbers is if they're in quotation marks when they come up rather than sort of standalone numbers. So if I did, let's go back to my sort of blank scrap sheet. Let's say I just ran the number 4. Here, if we go down, it says 4. But if I did 4, it's in quotes, it wouldn't be able to do math with that. So if I did 4 plus 4, 8, if I do 4 plus 4, oops, yeah, 4, it doesn't know. It's not treating that as a numeric argument because of those quotation marks. So it's just important to note if you're seeing numbers and they're in quotation marks, it's probably read them in wrong or incorrectly as text, as a character. And so it can be really helpful to double check that if you're getting kind of weird results. So I mentioned factor. Factor variables are going to be basically categorical variables. They can be either ordered or unordered. So ordered would be things like first, second, third year students. So things that the distance from the first group to the second group is meaningful. They come in an order. Unordered categorical variables, we could say in the example I have on the screen, you know, which borough do you live in? the answer, there's not really any meaningful, we might have encoded those as 1, 2, 3, 4, but that doesn't actually mean anything. It's just a placeholder for our analysis purposes. Whereas the ordered, typically the number actually does mean something, that 1 is more than 2 and so on. So insofar as they have numbers assigned, the number isn't mathematically meaningful. It's really a placeholder for group 1, group 2, and group 3, if that makes sense. So you can actually give these factor variables levels and labels. And one thing I'll note is that this concept is different than how it works in, say, a tool like SPSS. So just something to be aware of. We could just assign what the levels will be here. So we could say, for example, so in our data set, we might go back, and I'm just going to do the summary again. Summary NYC $gender. And currently it thinks those are numbers. If I do class, you'll see this class command coming up a lot, because this is how we can find things out. It thinks it's an integer. And in this case, if we were to go to the NYC Haynes data dictionary, we would see that that is not the case, that it is actually groups and not numbers, as opposed to something like age, which is a continuous number. In this case, they've just encoded things as 1s and 2s. So we might want to recode that, and we could say factor. We could use this command, and then take the data we want to take, NYC gender, and then we're saying how many groups or how many levels are there. So we can say there's a level 1 to a level 2. Something you might note is I've written out each group that exists in the form of doing 1:2. If, for example, it was not just 1, 2, 3, but it was 1, 3, 5, I could do that C 1, 3, 5. I've never actually seen that in real data. Typically they are 1, 2, 3, and so on. And you can just put the levels 1:2. But the important thing is you cannot just put levels equals 2, because it will think that the only group is the number 2. R will get confused and give you an error. So it can be a little bit counterintuitive. It's not saying that you can't just say there are 2 levels. You have to say the levels are level 1 to level 2. And if there were 3, 4, 5, level 1, level 2, level 3, level 4, level 5, and you could just do that with 1:5. We could also reassign them. So let's say we found 1 and 2 hard to remember. We could say use levels as a command. So the levels of NYC gender are being assigned for male and female here. And then when we look, it's relabeled them. But something sort of annoying for those of us who maybe have come from a background, say, using SPSS, is once those are reassigned, it's not going to read them as 1 and 2. It's not sort of similar with the labels and the variables that we see in a tool like that. It's sort of rewritten them. Another option for writing that is you could say factor, in this case I'm doing age group, levels are 1 through 3, labels youth, middle, and aged. There's likely a technical difference, but the practical experience is very similar. Having read about this a bit, it seems that it's sort of, you're telling it to look for these as the groupings and then to just call them this, which may be more of a technical than a practical difference. So you may see both in your work going through in R. Just something to be aware of for when we want to assign these to be factors. So this is relevant in that here we saw, for example, that it was reading in this as a integer. Often is the case when we're pulling in data from for example, a CSV spreadsheet where it's not entirely clear. It may interpret our values incorrectly. So here it's interpreting it as an integer, but really we needed it to be a factor variable. So we talked a little bit about true and false, or the logical variables. Something to be aware of is that these also have numerical values. So yeah, we'll talk about, there's a question about how could you, if you had the raw value, could you group them yourselves? You could indeed. We're going to talk about that a bit more as we go more into working with functions and working with both if statements and some of the filtering functions that are available in R. So we will talk about that, but not today so much. With regards to logical statements, so something nice to be aware of is that true is the same as the number 1. So I apologize, this font kind of looks like an L or an I, but that is a 1. False is a 0. So what this means in practical terms is that you can actually use these logical statements to run, for example, you could figure out how many meet a certain condition by summing the trues for a given condition. So you could say, this would actually go through, if we just said NYC SPH less than 50, go ahead and do it just for the sake of argument. NYC $SPH under 50. It's gonna go through every row. So it stops at 1,000, it cuts off. And if they're over 50, it gives us a false. And if they're under 50, it gives us a true. And so the thing that can be nice, so we can imagine just going down the column and saying true, true, false, and so on, is that we could then use this to figure out how many people are under 50. So if we just said some, oops, we can see there's about 949 of 1527. The other nice thing, we could also, for example, take the mean. And so it's saying, what is the average across the whole group, which is another way, sort of a short way of getting the proportion of people under 50. So this could be a nice feature to be aware of when you're working with R. You can use these logical conditions because they code as 1 and 0 as a really quick way to look at the proportions as well as to just see how many in a given criteria meet a specific criteria. So something nice to be aware of. So I'm going to sort of wrap up. I realize we're running close to the end of time. Before we go on to data structures, though, I mentioned that there's this command class. So class, and then you can run it on a column. You could run it on an individual cell. As we'll see later, you can actually run it on an entire data structure. So an entire, for example, if we ran it on NYC, it would tell us it was a data frame. We will post a link on Slack. The other option in the immediate term to get access, as well as on OneDrive. The other option too is if you go to that GitHub that we've been pulling R curls from, you can find the materials there as well. So they will be made available. So yeah, I'm going to sort of stop there as we're running out of time and sort of leave the last few minutes for any questions before on Thursday we're going to really dive into the data structures that are available. All right.